Good day, my dear postgraduate students. Welcome to R.A.T.S, Racing Against Time for Students in Pathology. And this particular picture shall be an exact reflection of our plight. We are chained down, racing for something against time. And this particular topic is important from the university question point of view, and it forms an important armamentarium in recent advances, maybe 23 or 24. Tensins in health and disease. Now, what are these tensins? We find that there are a plethora of topics thrown at us, which makes both the student as well as the examiner uncomfortable. Students at least write something, but then imagine the plight of the examiners. God only knows why these are being included. Because I find that you have got topics such as bioterrorism, artificial intelligence, amyloidosis, Tensins, as well as whatnot. And there is absolutely no connection between these. Each one of this has been studied by a panel of experts and then it has been published. Earlier it is a publication, but later on it becomes part of the text. Basics are basics first. I'll take you to the basics of the cell. I find that there is a cell, the cell membrane over here, and this is another cell, in between which I find that there is an intercellular junction, in which I have got all these terms: the tight junctions, the desmosomes, the gap junctions, and the adherent junctions. These four names you people shall and will remember. There are some areas called as tight junctions. Hardly anything can be passing through them. The next will be the intermediate filaments, the desmosomes, the tonofibers, which knit the cells together. There are certain gap junctions which help in the passage of material when signal permits. And there are also certain adherents from which you find the cytoskeleton can be supported and signals passed on to. Hope I am clear with it. Have a little concept of this. Spend a little time. It is worth it. This picture is not from the text. Again, I repeat, these are important. The tight junctions, desmosomes, gap junctions, adherent junctions, and actin cytoskeleton. Very simple. This we shall be having in mind. This is my age-old friend, the amoeba, which we would have been seeing, drawing in whatever way we like at school onwards. What happens is it puts forward a process of the cytoplasm, and then there is a drawing of the rest of the cell along with it. That happens because of some filaments which are capable of extending and contracting. This is exactly what is happening in your skeletal muscle, cardiac muscle, etc. The actin and myosin filaments. So this is one. It is literally an extension, then there is an adhesion, and finally there is the transfer of the movement over here. And I find that from one adhesion to the other it moves. The cell over here. An amoeba is a freelancer. It can move wherever it wants. This is a little more complex version of it, but still remember. These are the epithelial cells and there is something called as the intercellular or the collagen matrix that is supporting the cells. There might be some cells within them also, which will be having some binding factors called the integrins. So I find that there is some contact between these cells and how the signal goes and how things move, we shall see. This again is a diagram of the cytoskeleton. Cytoskeleton means, please remember these two things colloquially, the actin and the myosin filaments. Here, I am finding the plasma membrane. This is the other side of the plasma membrane. There is an endoplasmic reticulum. There are two types of this, the smooth and the rough endoplasmic reticulum. This I probably feel is the rough endoplasmic reticulum. The mitochondria, etc., all of us. Now, what is important will be the filaments that are lying between, which are called the intermediate filaments. And they get connected onto the microtubules to form a reinforcement for the cytoskeleton of the cell. Whatever be it, you find that a cell is predominantly made up of water, but then still it has maintained its beautiful outline by means of this cytoskeleton. This concept we shall again have. Now, there is something called the supramolecular linkage. This is where things come for us. This is critical in the organization of the cytoskeleton. Also, as I had been mentioning, there is some kind of a signal that is being transduced or transduction of signal takes place. And the topic for today will have various functions such as cell adhesion, migration, differentiation, proliferation, development of tissues, genesis of diseases. Imagine migration I can understand, but even to differentiate, you find that these kind of signals matter most. So I would like you people to kindly by heart this list. I have made things as simple as possible for you people. This is a very superb diagram. Do not get perturbed. Just follow what I say. The text is over here. You can read it at leisure. This is the extracellular matrix. And there are various things such as laminin, etc., which can be over there. And these are the integrins or the adhesions. And these will be getting attached to certain blue substances over here, which are the focal adhesions. And in this, we'll be finding the group of substances called the tensin, paxillin, talin, etc. And some of them have been expanded over here. Focal adhesion kinase, then mitogen activated kinase, extracellular signal regulated kinase, and CAS or the family of cytoskeleton. So these are the things. They help us in, or rather the cell in proliferation, differentiation, migration, survival, motility, gene regulation, and also the cytoskeletal arrangement. Almost a repetition of the previous slide, but it is worth. So from here you find that there is a signal that is being passed on, and this group of blue substances pick up that signal and activate the cytoskeleton. This much we shall remember. God only knows why, but we shall. If we do so, definitely you will score over your friends. Tensin. This is one of these substances which acts as a molecular bridge between the integrins and the actin cytoskeleton. So integrin is part of the cell membrane, and between that and the cytoskeleton, there are a group of substances of which tensin is one. So it is a phosphoprotein localized at a focal adhesion. It has got 4 members in its family. We shall see what they are. But then the major function of it can be taken as contraction, convolution, and the functioning of the chorionic villi. Remember these as 3 C's: contraction, convolution, and the functioning of the chorionic villi. 3 C's. And I said there are 4 members. Who are they? Very simple. 1, 2, 3, 4. So tensin 1, C1, then 3, 4, and C10. These are the things. And they are mainly localized in these areas: the heart, skeletal muscle, kidney, and lung will be for tensin 1. And tensin C1, that can be a glomerulus and the podocyte of the kidney. Tensin 3 can be in the placenta and the kidney. And 4 can be in the placenta and prostate. So not much. This group of organs or tissues we can remember: the heart, the skeletal muscle, the lung, the glomerulus, placenta, and then prostate. So this particular table we shall memorize. Imagine you are getting this as an essay question. There is nothing wrong in by hearting a small table like this. Now, the tensin has got 2 domains. Domains means regions. One is a cytoskeletal domain called the N terminus and the signal transduction domain at the C terminus. The cytoskeletal domain is composed of 2 actin binding proteins, namely ABD and P10. And the cytoskeletal domain is absent in C10. This is a little difficult, but if you can remember this much: cytoskeletal domain and signal transduction domain, more than enough. As I told you, these domains, they will be interacting with the actin filaments. Here again, the signal transduction domain is composed of SRC homology domain, SH2, and phosphotyrosine binding protein, PTB. The SH2 interacts with the PTB and causes a signal transduction. So there is a binding, there is a signal, there is a transduction of the signal, and then it acts upon the actin, myosin, whatever it is. Definitely you will not be able to understand this. But let us see whether we can unravel something. Always in such a complex pictures, try to trace certain elements that you know. For example, with great difficulty, I have found that there is something called the tensins. Is that clear? So tensin 1, 2, 3, and 4. The other things you'll forget. And before the tensins, there will be something called the protensin, myelocyte, promyelocyte. And the protensins are here. And these themselves are being activated by the proto-PTP, C2 and proto-SH2 PTP. So this we have already seen, the C2 and SH2. This is how it is getting activated. All the other things you need not remember. So there is a protensin, then the protensin activates or is getting converted into the various forms of tensins. The other things are, and various functions can be duplication, fusion, etc. So this is one picture, and please do remember each one of this is an extreme study that has been done by research scholars. It is beyond our purview. Even if you are going to sit and read, you will not understand. But this much we can. The C2, SH2, and then the protensin, the tensin. Tensin means there are 4 types. This much we can remember. And the adhesions. as I said. Now, these are the integrins. This is the cell membrane. And this is the extracellular space over here where I'm pointing out there. And just below the integrins, you find that there will be a group of substances which are called as adhesions. They are actually I mentioned as 7 or 8, but here as many as 60 or even more could be there. Every time they conduct a study, they will be able to unravel some. So here is our friend, the tension. So in this junction, if you can answer the other things or mention at least a few like vinculin, paxillin, talin, actinin, etc., then you are adding to your answer. So there is the talin, the tensin, vinculin, paxillin, SRC, focal adhesion kinase, protein kinase C. So these things we will be able to remove. And this is a very simple diagram. Diagrams like this will simply fetch you marks, carry the attention of the examiners. This is almost a similar list as we had mentioned earlier. There can be signal transduction, adhesion, migration, differentiation, proliferation, development of the tissues. Apoptosis is another one. It can help in the development of a malignancy. It can be used for the treatment of malignancy, and it can also help in the fallout of cells or apoptosis, genesis of diseases. So these are the functions of the focal adhesions, under which we have got the tensin. So functions of tensin, it is straight in front of you. You people will be able to memorize it. You are better than me. So the role of tensins in health and disease can be adhesion and motility, helps in the cell survival as well as in development of apoptosis, and the C10 and tensin 3 control the migration and the interacting with the integrin. This is another diagrams. I keep throwing diagrams, not that you people will have to remember everything, just to have an idea over here. Again, the same. You find that there is a cell membrane. Then there is an integrin which is attached to the cell. There is an extracellular matrix. Very simple. And to this you will be finding all these adhesions can be attached. Talin is mentioned, etc. And these will be activating the actin and the myosin. The other parts of it we shall not take. I took this diagram because it was important. And it is a myosin filament over here from a skeletal muscle. Such a diagram has been taken. So what are all the various things? You people can visualize. Forget the rest of my class. Please remember only this. If possible, you explain. Now, all the three of these are cells. Just follow this diagram. It is the most important one of my entire class. So there are three cells. I am seeing the nucleus over here. And the green one is also the nucleus. And I find that there are the adhesions that are dots. And there will be also the filaments which are supporting it on the cytoskeleton. What will happen is, in some conditions, there can be a breakage or the weakening of the cytoskeleton. So here you find that there is some kind of integrity. That is why you have got the word integrity. But here it is loose. So what happens? The cell can get detached. It can undergo an apoptosis, or if it is aggressive, it can go in for an embolism or a further development in the malignancy. Leave alone that. Let us see what is happening over here. So this is one. A technique that I have learned, which you people also can adapt in your classes. So this is the matrix component. All these lines are all matrix. Matrix is just a network. These are more rigid. They are the filaments, the actin or the myosin. And this is what we call as the integrin. It is given over here, the lines. These are the integrins. So the alpha and beta. need not go into it. But what I'm worried about is this rose colored or a pink cap that is over here and the blue structures, which can be the adapter proteins or the tensins, the hero of our class. Now, what will happen is in certain conditions, there can be, see what is happening over here, there can be a loss of these tensins, as a result of which you find there is no support to the actin or the myosin filament net, as a result of which there is a collapse that is happening. And these are all other components which can be used for it. C10 you people can mention. Sometimes these are all asked as questions. You should know at least in which group it comes so that you can use one for the other. And this is a summary of the whole thing. The tension is stabilizing. That is what you people are seeing here. There is an external growth factor stimulation that is over here, or an epithelial growth factor. The C10 competes with the tensin and binds to the integrin. And what happens is, whenever there is a weakening of the tensin or a loss of it, it helps in the cell migration and it promotes metastasis. This is a superb diagram which I took several minutes or even an hour to understand, to the extent that I can explain to you people. A beautiful one which you people, if you can explain this particular diagram, you can forget the rest of my class. So what is happening here? Detachment of the integrins, loss of contact with the extracellular matrix, that can be apoptosis. And the tensins and the downstream signals. Sometimes what will happen is they are used for therapy. Now, if you people can actually induce this and it is a tumor cell, there can be a detachment, there can be a destruction of the tumor cells, and only that is being targeted. So for targeted therapy, this could be used. This is an beautiful set of pictures again. It is a gastric carcinoma. In one, I'm finding that there is a close adhesion. Though it is a malignancy, there is some kind of orderliness in which I find that the tensin is still intact. But probably with further loss of it, when it is undifferentiated, see the chaos that happens. You find that the cells are loose, they become dispersed, and they infiltrate elsewhere. So this gene expression can be used to study the aggression of a malignancy. Tensins. Any number of times you people should be able to explain this. So a word about this paxillin is given over here, you can go through it. But please remember these are my friends. So the paxillin is over here, tensin I added to this particular cluster, vinculin is over here, facts I had mentioned earlier, SRC we had seen. So again, the same plasma membrane of the cell membrane, and these are the integrins. Externally there is an extracellular matrix. And there will be the transduction of the signals which activate the cytoskeleton, the actin and the myosin, or any other substance for that matter. So I hope things are a little clear for you people. Otherwise, only God knows what I feel inside. That will be the reaction of the student and the teacher alike when they see such questions. This is the best picture in my entire presentation. Very simple, easy for us to draw. So there is an actin filament, another actin filament. They themselves are being interconnected by actin. That forms the cytoskeleton. The cell membrane we had seen. Integrin also we had seen. And these are all the adhesions. Externally, we have got the collagen fibers and the collagen matrix. So this will be it. So when there is going to be a defect in it, there can be a weakening of the actin, collapse of the cytoskeleton and the cell. A nice cartoon that is there, all of you will be aware of it. This will be the fate and the state of the students during exams. You people know only this, but you don't know this picture. This is the same state of the teachers during those exams. Are you aware of the secret that the teachers ask for the question papers when they come as examiners, study it, and then come and correct your answers? It will be very much needed because it is not logical. As I told you, God only knows. These are a good set of references over here. If you people are going to become research stalwarts who are interested in research publications and so on, you can go to this and you can go to the back references of these from there on. But this is my strongest message. See what is over here. 80% of the exam is always based on the one lecture that I missed and one topic that I didn't prepare. I usually put it as a statement. The question that I omit will be my university question. So this is all a lot of pessimism for students who are very optimistic, who want to enjoy life. Please read this. Don't be upset because of what you can't do. Do what you can, the best. Live as carefree and optimistically as you can, because some people are not able to do that. Perhaps I also fall into that. I hope I can be as happy as you people and write whatever that comes to my mind. So this is the last slide. And I remember my pathology professor advising me, liver, read only the liver function test. Whatever be the question in liver, you can write. You find that the liver function is upset in this disease. And in order to confirm, the liver function test can be done and you write it completely. Logically correct whether it holds or not. But Tensin, I'm very sure that you people will be able to write it much better than your counterparts who have not attended this class. Hats off to the stalwarts who have done the research in it and to you people who have listened. Thank you.